All right, so chemical reactions. So I talked about catabolism and anabolism reactions in terms of ATP and ADP in the previous video. We're going to be moving here more into chemical reactions in terms of body, enzymes, and pH, and what that means for us. So in terms of chemical reactions, these reactions are really only occurring, so like catabolism and anabolism, are only really occurring when these molecules have sufficient energy to either form or break up. Activation energy and so forth, which is coming up here. But one of the key things here is molecules are under constant motion, which motion is a form of energy, so kinetic energy. Now, reactions occur when there's a sufficient energy to overcome certain chemical barriers. And so when they collide, sometimes this kinetic energy is enough to overcome that barrier for the reaction to occur. Now, one thing that happens here is while you think, oh, they're moving so fast, they'll hit one another, they'll break apart, they'll form bonds, willy-nilly, it just happens. One of the things that happens, though, is that electrons in that outer shell, the valence shell, repel one another as these molecules get closer, right? Because what happens is those electrons are a physical barrier. Electrons are physical things. They're not just a magical made-up positive, negative charge thing, right? They're physically there. So that's like the electrons are physically pushing away other atoms' electron shells, right? So, but you need to get close enough to either form a bond or break a bond through a collision. Well, how do you do that? How do you speed up these reactions, or how do you cause them to initiate, right? Well, the body has, God has created in the body proteins to speed up these reactions. What are they? What are these proteins? Anyone? Enzymes, right? So enzymes are a way that the body speeds up reactions, chemical reactions, to make things occur. Right? And the enzymes are used everywhere. There's an enzyme for, you name it, it has it, right? So, but why do we care about enzymes here? Well, what they do is they help reduce the activation energy. So activation energy is this number based upon the chemical bond types, specifically single or double or triple covalent bonds, or ionic bonds, or polar and nonpolar. It's the energy required to either form or break apart those bonds, right? Because everything requires energy to either make or break something. It's the way God created the universe. Enzymes reduce the energy required to make this reaction occur. So they are a catalyst, right? They speed up the reaction by reducing the amount of energy it takes to initiate the reaction. I'm trying to think of a good metaphor for it. I had one back in the day. Can't think of it now. I'll probably remember it in person in the lecture, so that'll be a fun thing to look forward to. But enzymes are really important to biology, really important to physiology, because all of these reactions that we'll talk about, especially metabolism or cardiovascular system, respiration, cellular respiration, utilizes enzymes. So it's really important to understand why we care about them. And one important thing to note here is enzymes are not causing a reaction to occur if the normal reaction cannot occur. And that's kind of a weird way of stating this, but if there's a reaction, say, one molecule A and B form molecule C, if that isn't supposed to happen because of energy requirements and so forth, an enzyme can't speed up that reaction to occur, okay? Enzymes are only effective when a reaction can occur spontaneously, is the word that we use, in terms of free energy available for the reaction to occur. So it will speed up a reaction that's naturally occurring, but it will not cause a reaction to occur if it's basically can't happen, okay? So that's an important thing to know about enzymes. Now we're moving on to kind of properties of water, mainly from the fact that water makes up 70% of humans by mass. So most of us are water. I joke with my biomedical engineering students, we are one non-Newtonian gelatinous blob of water. A lot of fancy words there, but yeah, we're just one big blob of water. So we really do care about the properties of water. In terms of physiology, it has a lot of great importance, especially in terms of stabilizing body temperature, right? Water stores a ton of energy. If you have tried to boil water quickly, you will know it takes longer than you think it should to boil water. Because there's covalent bonds in the water molecules, there's hydrogen bonds in the water molecules. Those can store a lot of kinetic energy, which is what heat is. So water has great importance to help stabilize homeostasis in terms of body temperature. The other thing it does is when it evaporates, it carries away heat, so it can help us cool down, hence perspiration with sweating, right? So that's one of the things that God created in us to help us cool down. It can act as a lubricant in our joints, so it's protection. It can act as a cushion in terms of mechanical functionality from protecting us from trauma. The mechanical term there is called a dashpot. If you're a car guru, which I don't know how many you are, if any, there's like an air suspension system. That's a dashpot model. It dampens the blow. It doesn't stop it, but it dampens the blow, and that's what water does, based upon the chemical bonds of water. It's involved in a lot of reactions. Either it's a product of a reaction or it's required for the reaction, called a dehydration reaction or a hydrolysis reaction. Especially when we talk about ATP and ADP catabolism and anabolism reactions, water's involved. So again, ATP, ADP, need to know it. Oh, jumped too far. Water allows us to mix things and carry things throughout the body, so it's a great solvent. It's also a great solute, right? A solvent dissolves something, or a solute is something that is getting dissolved into something else. Water can be both. It's a great molecule of life. It can suspend particles. So these are particles that don't dissolve and they're not disassociating, but they can suspend things, say blood cells in your plasma, i.e. blood, right? Immune cells, other things. It's a great medium to suspend and carry particles. It can also, it's a colloid, another way of saying it can carry tiny particles through a medium that don't settle. So like milk, plasma of the blood, cell interior, it carries tiny particles pretty well as a medium away for locomotion of those things. So water is of great importance here. Moving on quickly here, I know we're kind of jumping around, but I'm giving you the important things you need to know from this chapter here. Acids and bases is another one. So body, again, lots of reactions that occur. We have acids and bases that the body has to maintain homeostasis with, okay? We have ketoacidosis, which is the ketones, which is the byproduct of proteins when we metabolize them, can build up ketone bodies in the blood, which are from the kidneys, can cause the blood to become very acidic because they are an acid. Get too much acid in your blood, it starts destroying other molecules via protonation. It's a chemical reaction, stripping protons. Your body wants to prevent that, right? So it needs to create other molecules to balance out acids, which are molecules that a proton is donated, like hydrogen chloride, that releases the hydrogen ion. So like chlorine, and hydrogen. A base is one that accepts a proton. I know it's kind of a funny way of thinking it, but they act, protons are just, bases are just, more positively charged in a way, or they become negative. It's a really weird way of phrasing it, but protons become negative. So you have acids and bases. Are important. The strength of these are important in biology. Okay, so strong acids or bases really completely dissociate in water because water's polar. So you either form lots of hydrogen ions or hydroxide ions. Then if you have weak acids or bases, they partially dissociate in water, producing little amounts of hydrogen ions or hydroxide ions. The dissociation of these is reversible in both, but as in you can make the original molecule is what we mean by reversible. But if it's strong or weak, that really depends upon whether it's easily reversible or not. Basically, that's stating how much energy it takes to reverse the reaction in terms of making the acid or the base. And I have this table here to provide for you kind of very common acids and bases. Water is both an acid and a base at the same time. So water can form a hydronium ion, so it's positively charged because it accepts a hydrogen, right? So it's becomes an acid here, highly reactive. Or it can lose a hydrogen, right, and becomes a hydroxide ion, which increases its base, right, because it's going to want to accept a hydrogen later on, versus an acid wants to give up a hydrogen, right? So that's probably the easiest way to understand what an acid or base is. Like an acid wants to give away a hydrogen or a positive charge. A base wants to accept a hydrogen charge. So it's going to be more negative than positive. Again, some of these things you just have to memorize. Because of this acid and base scale, we have what's called the pH scale, which we all know. So the pH of something ranges from like 1 to 10. Or in this, not 1 to 10, 0 to 14, excuse me, because we're in a different base here in terms of math. But neutral is 7. Basically, pH is just the concentration of hydrogen ions in our solution. So for us, how much hydrogen ions are in water. So neutral is 7. So it has equal amounts of hydrogen and hydroxide ions. So basically balanced out acid and base. If you become more acidic, that means you have more hydrogen ions, right? So you're less than 7, so your concentration of those go up. Now, when you're basic or alkaline, that means you have more base than acid or more hydroxide ions than hydrogen ions, right? So there's less free-floating hydrogen positive charges. In terms of why we care, well, in household items, this is kind of an example of this. Sodium hydroxide, basic. Bleach, pretty basic, right? You would think it's an acid, but it's not. It's really a base. Blood, you can see here, is about 7.4, so it's more on the basic side than the acidic side. Mainly from the fact that the body wants to be more basic than acidic, because acid is more corrosive, more damaging than basic. Okay, saliva is more acidic than blood because that actually helps digest food. So it's 6.5. Then you have coffee, which you either will love now or will love in the future because, you know, you're nursing students. You will have late nights or all-nighters or back-to-back all-nighters. Very acidic. Tomatoes are acidic. Vinegar and obviously lemon juice. And stomach acid is very acidic, right, to help break down foods. So that's kind of examples of basic things you would find that fall on the pH scale. So why we care about pH again, it's because of blood really, as well as other chemical reactions in the body. Talk about digestive system. Normal pH 7.35 to 7.45 in the blood, right? We go below this number, acidosis. Go above this alkalosis, right? So it's a very tight window here for homeostasis, right? So the body, this is another thing that it's trying to maintain here, is our pH in our blood and our body, right? So it's a very tight window of what the body needs to maintain. So there's a lot of reactions, a lot of enzymes, a lot of molecules in the body to balance this out. Hence, where when you come to salts and buffers, these are the things that are going to be balancing out the acids and bases. So a salt is a molecule or compound consisting of a cation, so a positive charge, with an anion, right? So other than a hydrogen ion is what we're trying to sample here. So that's what a salt is. Example of a salt that's other than the hydrogen and hydroxide is sodium chloride. So sodium is positively charged, chlorine is negatively charged. Sodium bicarbonate, kind of same idea. A buffer really combines kind of the acids and bases pairs that can come from salts to balance out that pairing of the positive charge and the negative charge, or removing the concentrations of the hydrogen ions or the hydroxide ions out of the body's solution. And we call these kind of pairs conjugate pairs of acids and bases, right? So a conjugate base is really what forms when an acid loses its hydrogen ion, right? So hydrogen floats away because it gets dissolved, right? And then it becomes basic, right? It has that negative charge because it's lost that positive charge. Same with an acid. The conjugate acid is formed when a proton is added to that base, right? So we have an acid. The third point here, we have an acid, we have a base. It can go back and forth between a conjugate base, which is related to the acid, and a conjugate acid, which is related to the base. So in terms of when we have a lot of an acid solution, right, a lot of hydrogen, a conjugate base accepts that free hydrogen to counter the charge, to counter that change, to decrease the free concentration of the hydrogen ion. Now, if we have too much base, the conjugate acid will release the hydrogen ion to release and produce more hydrogen concentration in solution to balance now that negative charge that's just come in. So buffers are really just there to either release or accept hydrogen ions to balance out the positive and negative charge, right? Because when we're talking about pH and acidosis and alkalosis, really we're looking at the concentration of the hydrogen, and we want to maintain that between 7.35 and 7.45. You're trying to maintain the free-floating hydrogen ions in solution, right? That's what pH is how much hydrogen we have in solution. So examples of buffers, so bicarbonate, phosphate, and proteins are also buffers, right? To kind of balance out the hydrogen ions and the bases and the acids, right? So an example of this, when an acid is added to a buffered solution, right? A conjugate base is formed from the buffer to remove the free-floating hydrogen ions, right? So this will reduce the number of hydrogens free-floating in the solution, reducing the pH. Or excuse me, increasing the pH, right? So it's the inverse. Increase the pH, less free-floating hydrogen. Decrease the pH means more free-floating hydrogen, right? The chemistry is weird. One of those things we'll just have to memorize. We'll end this part here. And we'll go on to something else later in the next part.